Okay, well, good morning, AZ4H, and I want to welcome you to our final day of our Science and Ag Week presentations. I'm quite excited today because I get to host a friend and colleague of mine, Dr. Jerry Lopez, on his specialty of food safety. I'm actually really excited because as much as the two of us have worked together, I've not actually been able to watch him teach. So this will be a treat for me. I want to share with you that Dr. Jerry Lopez is a specialist with the University of Arizona. He works in food safety, water quality, and the transmission of pathogens in indoor environments. And he's also our Arizona 4-H STEM specialist. So all of the other STEM programming that you've been engaged with, if you've been to our camps on campus or our virtual camps, those are headed up by Dr. Jerry and his team, and we're so glad to have him with us today. So today we've got food safety as our topic. We're going to break it up into 3 sections, and I'll turn it over to Jerry after I go over just a few notes on the tools for the presentation. So if you've joined us before, you know that you have option of using the chat to share information. So if there are questions for you to respond to, use that chat box and type in your answers. If you have questions for Dr. Lopez, you can type those in after you click the Q&A icon, and then those questions will sit there until we resolve them. So we may get to those at the end of the section or at the end of the presentation. If there happens to be any questions left over at the end that we don't have time for, we'll find a way to get that information out to you because we want to make sure you're as informed as can be. You don't have to worry about background noise or background image because this is a webinar feature, so we don't have you chiming in vocally or with your image. So don't worry about yelling at your dog if it starts to bark behind you or telling your little brother or sister to get out of the room. They're welcome to peek over your shoulder and check in. We won't notice them. And with that, I will turn it over to Dr. Lopez. Thanks for joining us today, Dr. Lopez. We're looking forward to it.
Absolutely, my pleasure, and thank you for including me in this program that you put together, that you spearheaded, Dr. Morris. So I really appreciate it. Yeah, this is going to be interesting. You as a former science teacher, this is going to be neat. Let's see how many times I say So first time. I'm really glad to be here, and I'm glad that you all have been able to register for this session. The title of our presentation, the first one at least, is The Field of Food Safety. What does all that really involve and entail? And this nice picture graph of a lot of different terms, provided by the FDA really sums it up, right? Smarter food safety. Some of the topics that we're going to be going over is going to be foodborne outbreaks. That'll be the first item that we tackle. Second one is I wanted to provide just a little overview with some images that a undergraduate student, Elena Rivas, and I put together on culturing bacteria, so you can get a sense of some of the items that take place when you're working with food safety, at least from the cultural perspective. And lastly, we're going to take a different shift and move into giving you a presentation on some of the links that are available online, specifically for the College of Agriculture and Life Sciences, in order for you to begin to explore different careers in our college. So starting off with food safety, when you think about that, you're probably thinking, well, what are the different pieces? What are the different moving parts to this? Who's working on the different aspects of trying to address a foodborne outbreak? And as all of us understand and can relate with the COVID-19 health crisis, we sort of understand in the background there's a lot of moving parts. And as we go through this food safety foodborne outbreak presentation, you can make a lot of connections to how this would be applied to an infectious disease outbreak like we're currently experiencing with COVID-19. So take a second and sort of look at the different organizations that are sort of playing a role in this. You might think, well, if you ended up eating something and all of a sudden, or several days later, or all of a sudden you end up, feeling sick, your stomach's not feeling good, you might even have to run to the restroom, have diarrhea, different symptoms might result. And you might, if it doesn't get better, you might have a feeling your mom or your dad might end up taking you to a clinic or a hospital, right? That might be the first part. So they definitely play a role in addressing these. Samples are taken, and that's where the epidemiological aspects begin. Especially they'll take either some samples and determine what type of illness it might be. And if they start grabbing or obtaining a lot of different cases or individuals coming in, then that red flag goes up. So part of that then is also the Arizona Department of Health Services, the public health department, right? They then start getting involved in addressing that issue and collaborating and discussing, working with the hospitals or the clinics. And depending on what's going on, the private sector might be involved in order to address that in terms of taking the testing that's working with the hospital to actually have these samples tested, as we are currently seeing with samples being collected and tested then for the COVID-19 virus. Now the USDA, when we're talking about a food safety issue, a foodborne illness outbreak, the USDA arm is definitely involved, and so is the FDA and the USDA are involved in those different aspects with different lenses, but they're addressing that actual issue, the rise in illnesses. And of course, you have academia, us here with the University of Arizona, you have the other universities across the state and across the country that are involved in actually collaborating with ongoing projects that might be existing. We have some projects, our department, there's several food safety individuals that do different types of projects looking at different types of organisms and prevention measures. And then lastly, but not least, really the hub of all of this, as many of you have noticed with the COVID-19, is the CDC, the Center for Disease Control and Prevention. So I bring all of these up because there's so many different career opportunities for you to explore. And at the same time, you can see the complexity of how it is to address a foodborne outbreak. Okay. So where do we go from here? Where does it occur? So in the fruit production, right, obviously. Let's see here. So it could be at the raw commodity level in the processing of our food. It could occur in the food industry. Right?
He's watching out. Oh, I found the guy with the drum gun.
The food industry of the actual preparation of meals, grocery stores. It all depends where the contamination actually took place. And then lastly, our home, right? That's a major thing in terms of secondary transmission at home. Someone becomes ill, and then because of the importance of washing our hands, using kitchen cloths, or cross-contamination when we're cooking, or not rinsing our fresh produce, or cooking our meals to the right temperature, there's a potential for an outbreak or illness to occur. Now, the question is here, what causes an outbreak, or what causes the actual illness? And I'll give you a second for you to sort of think about that. So you probably guessed it. There's definitely viruses can play a role. Bacteria, that was probably one of the ones that first came to your mind. And of course, parasites, one of my favorites. So different organisms based on what their characteristics can play a role in contaminating our food and water, and then we become exposed by taking that in, eating it or drinking it, and that's how then after a period of time we then become ill. So something that I want to explore, so now we understand what they are. Before we can determine if it's a virus, a bacteria, or a parasite like cyclospora, we have to detect it, right? And so what methods do scientists, food safety scientists use? Well, there's 3 different main categories, right? We have to first collect the samples, and we'll be doing an activity just to sort of give you a sense of what's going on here. So in the case here, there's a water sample that's been collected. Here we got some ground beef, and if you can notice the Petri dishes there. And then here there's a swab that's going on. So just like in our case with COVID-19, they do either a throat swab or a nasal swab. Here you can use swabs to sample surfaces. Very similar type of process. You have to do it aseptically, carefully, and then that's transported back to the lab. Now, once in the lab though, there's different tools that can be used. Microscopy is one at certain points. The cultural aspect, particularly with bacteria, using specified types of agar plates, media, growth media, and then finally the molecular. And that's really an area that a lot of our technology is moving towards. And not only just detecting little segment or a marker for that particular pathogen, but actually doing the entire genome. And so as you look at this one screen, industries of the future, the actual workforce in the coming years is really going to revolve around these aspects, and then all the different supporting roles that go with this from the epidemiological perspectives and so forth. So I want to transition into some examples for you to be able to put this in a context. So this example here is a parasitic example. Cyclosporal cayentinensis. Our research group focuses on this. Over the past few years, there's been an increase of these incidents. And what's interesting, as you can see here with the graph below, is that there happens to be a season that it becomes really, really evident and the cases rise. And that sort of happens within May, June, May, early or late May through late June, August. And sometimes it extends during that time. There are cases that occur throughout the year, but they're more sporadic. And here you could see the cases that are identified by different states. The health departments are notified. The CDC is tracking this information as they are currently tracking for the COVID-19. And the industry that's involved really ends up getting impacted by these at times. And there's a whole long line of companies and producers, that entire chain that's impacted. And so the epidemiological study, experts, folks who study these different types of fields or variety of fields, use their skill set in order to be able to do that traceback analysis. Just quickly, we have scientists. In order to be able to understand that pathogen, you need to be able to understand the specifics, the actual characteristics of those organisms. And in this case, it being a virus, it being a parasite. And you can see these little sporozoites are the ones that actually end up infecting our tissue, our epithelial cells in our intestinal tract. So another example here is going to end up being E. coli 0157H7 that ends up being a major problem at times, right? And so you have here that the graph is showing the cases that are occurring. And this was a major outbreak, 36 states, 96 hospitalizations, and unfortunately 5 deaths. Now, lastly, here's norovirus. So this is one of the foodborne viruses that is one of the major causes of outbreaks, diarrhea and vomiting. And so if someone is sick, it really transmits very easily. The viral load that someone is shedding is actually, through their, when they go to the restroom, potentially through their coughs or through their mucus, their sneezing, getting on hands. And it gives you a sense of the different ways that it could actually be transmitted. So finally, making a full circle back to this, you've seen the different components that are involved. So when you look at all these different agencies, these partners that are all working together in order to address an outbreak or some type of infection or disease that's happening in either isolated communities within a state, or as you saw in those graphs, it's actually can spread because the commodity, that product, whether it's lettuce or ground beef or a vegetable, a fruit, whatever it might be, is being then transported and delivered to multiple locations, to different facilities, not only to restaurants, right, but also then to supermarkets, and humans then are being exposed. So it's really important to be able to identify that and prevent that outbreak getting worse. And so again, these are all different career opportunities that someone is able to pursue. And if you like more the science aspect, the deep science piece of it, there's careers that focus in that. If you feel like or you enjoy working more with people and being out and meeting folks, there's different positions that sort of are tailored that require those type of skill sets. So, and lastly, I wanted to go ahead and add this slide because it gives you the depth and the breadth of this, the career opportunities. PulseNet USA is an actual organization. It's a team within the government agency, the FDA, the CDC. There's different groups that are working together, and there's actually regional areas that they focus and monitor when these outbreaks occur. And so these offices need people, everyone from communication, the business aspect of it, leadership position, administrative leadership positions, and of course the scientists and different variety of those type of scientists. So there's opportunity for you to be able to travel across the country, as well as internationally. With the COVID-19, we all realize that there's a value in working together and being able to collaborate with our partners across the world to address these outbreaks. Okay, so before we move on to the next section, I wanted to ask Nick, if there's any questions.
I can't talk, but I'll.
Yeah, you can go ahead and actually put a question. If you can type a question out, that would be great in the Q&A.
We don't have any at the moment, Jerry.
Okay. All right.
I would say if anybody wants to, if you think of something as Dr. Lopez is talking about the next topic, feel free to throw it into that Q&A anyway, and we'll get to it at the end.
Excellent. Okay, so now this part right here is just to give you a sense of the different things that you work with in the lab. Now, this is dealing with just culturing bacteria. And so if there were some scientists, like let's say there were some spinach that was involved, or in a bag, mixed lettuce bag with either carrots or cabbage or whatever it might be, that's part of that medley, and there's suspicion to assume that that particular product could be associated with that outbreak. And so back in the laboratory, scientists then would start going through the process of identifying if that's there. And so this is an example of culturing where we would be looking for bacteria. So one of the nice things is, and one of the things that really sparked my interest in science and pursuing a career in that and majoring in that, was that you're able to work with your hands. And at the same time, there's times that you're working with your hands, there's times that you're reading to try to understand the methodologies or analyzing the results. So there's a nice combination of that. So one of the first steps would be then is to actually tear, that's what the term is called, to zero out your scale here before you weigh out your spinach. And some of you have probably done that in your lab classes. So you then continue, most of the time, it would, depending on what the commodity is, the product, there's about 25 grams that are measured out. And again, aseptically, because you don't want to contaminate that. You then would place that, transfer that sample into a bag. And this could be an activity that you may want to do at home when you're feeling or have the interest of participating or doing something like this. This could be a certain way how you can go ahead and approach it. So you would try to weigh out using a cooking scale, a certain amount of grams of that particular, whether it's, fresh produce, or maybe it's some ground beef or some cheese, whatever it might be. And then you transfer that to a bag, a Ziploc bag. And again, without using expensive pieces of equipment or supplies, you can use the type of supplies that you have at home, but just be careful to make sure that you're not contaminating, cross-contaminating with your hands. Make sure you're washing your hands. If you have gloves, wear gloves, and be precautious. So the next step then would be is you would add some water. So now one of the things to keep in mind is that you don't want to go to your faucet, your tap water, and just fill up your measuring cup and then add 40, 50 milliliters of water, because as you might know, there's bacteria that's found, heterotrophic plate count bacteria that's just naturally there. It's not harmful to us, but obviously that's going to end up contaminating that. And when you culture that, your Petri dish is probably going to be full of things. So you may want to boil some water first, kill whatever is in there, that becomes sterile, and then measure that out. And if you're waiting for it to cool down, make sure that you cover it with something so that it doesn't get contaminated with things that are floating in the air. Once you've done that, you want to do about 30 seconds to a minute, just slowly, manipulate it in your hands. Make sure that it doesn't, leak out of the zip zip part of the bag. And we would use something else, a different type of stomacher apparatus, to actually carry out this type of process. Next thing is these Petri dishes. I'm sure on Amazon, there's probably some, or anywhere online, some inexpensive kits that you might be able to buy, some auger plates and TSA, some generic type of plates that are really susceptible and open to culturing or growing multiple types of bacteria would be the kind that you'd like to use. So as you label your plate, you want to make sure that you always put the date, your initials, what type of product that you're working with. So there's information on there. So after you come back and from putting it in the incubator, or in your all's case, in your garage or someplace warm, you can then know what the different plates actually are from. And so the next step then, using a little, transfer pipette, something, again, these items are available online, something that you can actually pick up a little amount of volume of the liquid that's inside the bag. You would do that. And the plan then is to transfer it over to the actual Petri dish, the auger plate. And if you notice, make sure that you don't put it on the plate that has the cover, but instead on the side that actually has this little jello-like agar substance. That's food and nutrients for the bacteria to be able to grow on there. And lastly, this is a little hockey stick that we like to refer to it as, and it's to actually spread that liquid that you've placed on there. Okay. And following the spreading of the actual liquid on there, it should end up being nice and not necessarily dry, but spread out evenly. I mean, you might have to do that a few times until it actually then thins out over that. You don't want to put too much of that liquid on there because then it's not going to end up be able to dry. But in case that you do, just make sure that it might take a little bit longer for it to actually dry. Lastly, this is going to be one of the challenges here with step 11. Here in our laboratory, we would put it in an incubator, and it would incubate for 18 to 24 hours at 37 degrees Celsius. It's a little, warmer, so you might, I was thinking about this the other day, and putting it in the garage, particularly in the summer, might be a perfect spot for it to actually grow and incubate. So finally, after that, you would come back, you would look and actually see what's actually grown on there. So it gives you a sense of a fun activity and, to do at home. And you can actually do this with a lot of different substances, right? If you get some Q-tips and you have some sterile water again, and you want to take samples of different things, you can really explore and think outside the box of different ways to utilize these type of methods. So this pretty much ends this part of the presentation. I'm not sure if there's any questions, Nick, before we move on to the next.
Dr. Lopez, I don't see any questions in the Q&A, but I did want to ask, is it readily available for just anybody to get some of that agar material to be able to do this sort of thing? Can they find it online or through Amazon or something like that?
Yep, absolutely. It's definitely available online, and just checking the prices. It all depends the amount that you end up actually getting, right? Either the cases or the quantities of those, and really just getting the least expensive ones and starting to play with it. These already prepared is the best way to go. If
okay.
I do, say that again?
Oh, it's just, okay, that's good to know that it can come already prepared.
Yep, they sure do. And if there happens to be, one of the agents that want to actually explore this even further, we can work together and maybe even prepare them in our lab and coordinate getting these to a group of kids that actually want to explore this. So this would be a fun thing to actually end up doing. We normally do these activities. We have a handful of these type of events over the course of every year that we've been doing for the last few years, where kids actually come on campus and they visit our lab, and within an hour or so, we're able to go ahead and go through that process and actually do it on hand. So we can have the ability of doing that in our lab if you want to visit the main campus at the U of A, or we can coordinate and do something this at your county level or tribal community, federally recognized tribal extension program, for those of you FERTEP agents out there that would like to do something like this.
Oh, well, that's a great activity, and I'm glad to know that kids can do it at home. I think that's a really cool practice. So this will be a nice step, and maybe what we'll do is, even if it's just a link to some prepared slides that you think would be sufficient for somebody to do this, that way they know what they find online matches what you would recommend.
Perfect.
That sounds great.
Excellent. Well, we can go ahead and then transition into our last presentation or last portion of the third topic. And this was actually exploring, or how to go about exploring the variety of different majors within our college, the College of Agriculture and Life Sciences. And the nice thing about this is that you'd be able to explore other opportunities as well. So I think at times you sort of might feel that it's overwhelming, you're not sure where to start. And so I took the time to look up and provide you all with this wonderful link right here, degreesearch.arizona.edu. And this is an excellent search area that you can search the U of A degrees. And if you notice here, you could either search them by all, right? Or you can select the college that you might be interested in. And so the first one there is the Agricultural Life Sciences. So I went ahead and I selected that link. And then this other image here found 34 UA degrees in the College of Agriculture and Life Sciences. And if you notice, it's broken down very nice and neatly. This title or abbreviation CALS stands for the College of Agriculture and Life Sciences. And so you can actually go through each of these different tabs and identify what are the requirements, what are most of them have all 4 years of the classes that are needed to be able to complete that degree. So it's a really wonderful site for you to be able to go to and inform yourself as you begin to prepare and plan and discuss with your siblings, your parents, your grandparents, the different opportunities that you're considering. And the more you understand and are aware of these different majors and the specifics of those, the better informed that you're going to be able to make your choices as you start moving through the plans of graduating through high school and then applying to a community college or directly to the University of Arizona. And if you notice, here's the food safety tab. So that is a wonderful piece of information right there. And so the resources are excellent. So now, when you think of the different majors that are possible within our college, I'm going to focus on this for our college, but you could do this with all these different majors or different colleges. So we have traditional agricultural type of majors, commerce, we have health professions. Part of what we were discussing with the foodborne outbreak really, sheds different or overlays different areas, right? And so that's really interesting. And I may also add that as you're exploring these different majors, some of them require a minor, or maybe you want to take some electives that would be perfect for you to take them in another college. For example, the College of Public Health, that might end up having some epidemiological classes or risk assessment classes that you may want to complement what you're learning in our department. I'm in the Department of School of Animal and Comparative Biomedical Sciences, and these are the majors that we have. So, and then these others, helping professions is another one, science and technology, and then lastly, the sustainability. So we'll briefly touch on these as we go through the rest of the slides. So for those of you that are really interested in traditional agricultural, we have some major, interesting areas that you can explore and pursue: animal science, sustainable plant systems, agricultural technology, management, and education. And something that might be coming to your mind is like, wow, what exactly is sustainable plant systems? And here's an opportunity, again, going to that link, degree search, you're able to actually select that major, and there's a lot of information there that can actually guide you and start answering a lot of these questions. Commerce. You never thought that there might be some commerce or business-like courses in our college, but there actually is, and they're really, really interesting. And so, you can see everything from environmental water resource economics, agribusiness economics, retailing and consumer science. We have the environmental science, family studies. So there's definitely a lot of different areas that fall within that focus point. The health professions, as we discussed earlier, everything from veterinary science, nutritional science, animal science, microbiology. And something to keep in mind now that the first cohort, first class of the School of Veterinary Medicine is going to be starting here in the next a month in August. This is a really great transition from you attending the University of Arizona, deciding on a major, for those of you that are interested in pursuing vet school, being able to meet the professors and earning your actual degree and applying to the U of A and some other vet schools. But it's a really exciting time with the vet school actually starting here at the U of A this first class. Now, we've gone over these first three: traditional agriculture, commerce, and health professions. Now we're going to focus on these other ones: helping professions, science, technology, and sustainability. So helping professions, what exactly does that mean? So we have the agricultural education program. That is a phenomenal program. There's some great professors there, and the work that they do are really exciting. It's agriculture education for teachers, high school science teachers, and a lot of other areas that these type of degrees can prepare you for. And the family studies and human development is also a really interesting and important major for those of you that are interested with that, with social work, criminal investigators, the counseling, student services. So child and family advocates. So again, as you continue to inform yourself, go to that website and try to have a better understanding of what you might be interested in pursuing, these sites, that website and the different tabs is really going to give you the information to try to figure out, pursuing this type of major versus this other type of major. And let me go back one. Science and technology, here again, microbiology, the plant sciences, agricultural technology management, biosystems engineering. The industries of the future are really going to be cutting edge in terms of being able to solve the current problems that we have and problems that are in the future. So the professors and graduate students are in that realm of actually trying to address those challenges and solve those problems. So it's really exciting that, College of Agriculture and Life Sciences really encompasses a lot of different majors. Lastly, sustainability. We all know the importance of taking care of our planet and our resources. And, some of you might be interested in actually pursuing really specific degrees that focus on that. So you are our future. Seize the opportunity. These are some resources that are there for you. This is someday we'll be back to being able to have our graduation ceremonies in person, and you might decide to pursue your degree at the University of Arizona. And here at this photo here is a photo of our lab people that have been with us in the last year or so. And so it's really exciting. A lot of these are undergraduate students or graduate students, and actually staff that are part of our team. So thank you for having me. So this concludes the last part of our presentation.
Well, Dr. Lopez, I want to tell you that was a great presentation. I always love learning more science stuff, and I love the activity you provided us, and I think this overview of the majors and careers that are available is fantastic. So thank you for participating with us and sharing your expertise, and I'm really looking forward to continuing to develop new opportunities for our 4-H family around the state of Arizona as we continue to develop new ways of connecting with our kids and our staff and our volunteers. So thanks again for sharing your expertise. I don't see any questions in the Q&A box,
Okay.
but I will let our attendees know that they can certainly send questions in to me. I think my contact was on the registration, and I'll be happy to forward those along to Dr. Lopez, and we'll get answers to you if you think of something after the presentation today.
Perfect.
So thanks again, and just as a summary of the whole week, those of you who have participated each day, or those of you who have just kind of jumped back and forth, our hope here this week was to provide you an opportunity to hear about how science concepts and skills and strategies are used in problem solving in agriculture. And I think today we've got a great example of that objective, and I'm so happy that you were able to participate with us this week. So thanks so much for all of our attendees, and those of you who will see these videos in the future. Stay engaged, stay sciencey, and keep developing those skills, and we look forward to engaging with you in our next opportunities. Thanks a lot, and have a great day.